Hello, my name is Austin Carlson, and today I'll be presenting barium ion sensing with commercial IPG potassium molecular probes. First, why is it important? Fluorescence imaging of potassium ions is significantly relevant within biology. Potassium is the major contributor to the electrochemical gradient within cells and is therefore necessary for neuron firing and muscle contraction. In pursuit of studying these dynamics, a number of highly selective and sensitive molecular probes have been developed for potassium, including the IPG family seen above. Detection of barium, also known as barium tagging, would enhance nuclear physics searches for neutrinoless double beta decay. This is a theoretical process in which two neutrons decay into two protons, releasing two electrons and two neutrinos. The neutrinos then annihilate one another. The ionic radius of barium and potassium are extremely close, within about 3 picometers, meaning highly selective potassium sensors also tend to be selective for barium. We aim to characterize the IPG family's ability to act as barium sensors. First, results are presented from bulk fluorescence spectroscopy studies. On the top, you'll see the emission studies of each molecule, with excitation bands around 525 nanometers and emission bands around 545 nanometers. This is to be expected, as the molecules have equivalent fluorescence groups. On the bottom, you'll see selectivity studies for each molecule, with barium, potassium, sodium, calcium, lithium, magnesium, zinc, and cadmium. You'll see that, as predicted, each of the molecules are highly selective for barium, with IPG2 standing out above the rest as being selective for barium and no other metal. Therefore, we selected IPG2 for further studies. Then, IPG2 was used for further studies into single-ion microscopy. Slides are prepared by spin-coating IPG2, barium, or a combination of both onto them before imaging. In the first figure, on the top and bottom, which are the IPG2 and barium controls respectively, some fluorescence activity is shown. However, in the middle, which is the combination of both, fluorescence is highly enhanced as expected. In the second figure, you'll see the sensor returning to the off state after about 12 seconds compared to the control on the bottom. This is to be expected, as this is a characteristic known as photobleaching and is further demonstrated in the third figure. Each dot is an individual ion, showcasing IPG2's ability for single-ion fluorescence. And the time studies on the right showcase this photobleaching behavior with a steep drop-off after a number of seconds. Then, computational analysis of the IPG family was completed to reinforce our findings. To accomplish this, first, a conformational search was performed using XTB-CREST on IPG 1, 2, and 4. The lowest energy conformers of each were then optimized using density functional theory, commonly called DFT, with the B3-lip 6311G-D level of theory. This revealed the lowest energy geometry of each molecule, as seen above, in the gas phase. To investigate the fluorescence mechanism of IPG2, we used TD-DFT, or time-dependent density functional theory. First, we re-optimized the molecule in solution as it would be for our previous studies. Then, we attempted to simulate the UV-Vis spectra using TD-DFT. However, we ran into a unique error. We could not find this error in previous literature, however, so we began screening additional basis sets and functionals. None of this provided clarity, however, and later we found out that by simulating more excited states, we could sometimes get around this error. This then led to another difficulty, however, as when we added the barium ion to the complex, the geometry often collapsed completely. This could be avoided by using the DEF2 TZVP basis set specifically for barium, which provided a more accurate geometry with the ion. Then we were able to complete our simulations of the sensor with and without barium. Simulations without barium showed excitation that was quenchable as expected. Simulations with barium showed a drop in energy of the HOMO orbital, allowing emission and pointing towards a PET mechanism. This is demonstrated on the right, where excitation in IPG2 without the ion goes from HOMO -1 to LUMO, and then the HOMO electron can drop down into the HOMO -1 orbital, quenching fluorescence. In IPG2 with barium, however, this HOMO orbital drops in energy, leading to excitation from HOMO to LUMO, which can then not be quenched. In conclusion, the IPG family's strong selectivity and sensitivity for barium was demonstrated through bulk fluorescence. Then, their suitability for single-ion imaging was confirmed with single-ion microscopy, where we saw photobleaching characteristic of a fluorescent sensor. Then, computational analysis with DFT revealed the lowest energy geometry in gas phase and in solution. Then, with TD-DFT, the fluorescence mechanism of IPG2 was investigated and pointed toward a PET mechanism. We'd like to thank the NSF, Welch Foundation, and Department of Energy for funding this research, as well as Ion Biosciences for supplying the IPG dyes.